Mesothelioma Applied Research Foundation. Here we'll be speaking about the mesothelial cells. Well, Professor, let's get started.
Thank you, Dr. Maeda, and ohayo gozaimasu. Now, many of you will know a little bit about mesothelioma, or lots about mesothelioma, but many of you may not know very much about the mesothelial cell. So I'm going to talk to you today a little bit about this cell, and in particular, what happens when healing goes wrong, or the normal healing process, and then how things can go wrong. So the mesothelium is an extensive monolayer of specialized cells that line the body's serous cavities and internal organs. It lines the three serosal cavities, that being the pleura, pericardial, and peritoneum, and it's derived from the embryonic mesoderm. So that means that these cells are mesenchymal cells, but they have epithelial-like characteristics. And in these two images here we have, this is a testis, this is a mouse testis actually, the seminiferous tubules, tunica albuginea, and on the surface are a thin layer of mesothelial cells. This arrow is pointing to the nucleus of one mesothelial cell. Very hard to discern the actual cell, but if we look at it by scanning electron microscopy, what we see is a carpet of microvilli, one of the characteristics of mesothelial cells. This is a transmission electron micrograph, and what this shows here, and what I want to point out, this arrow is pointing to the basement membrane beneath the mesothelial cell. So you can see it's a very thin attenuated cell with microvilli on the surface. Now this will depend, of course, on its location in the body, because in some areas, such as the diaphragm and on the omentum, these cells are a little bit plumper. But in a lot of areas, they're very thin, long, attenuated cells. And I want you to pay particular attention to what we see. This is in the tunica albuginea. We have these fibroblast-like cells. This is the nucleus of these fibroblast-like cells, amongst a lot of matrix, predominantly collagen. And again, depending on the location in the body, there'll be a different amount of matrix and a different number of these cells. But just remember these cells because that's important for things I discuss later. Now, what do mesothelial cells do? We all thought that they were very basic cells that just provided a slippery, non-adhesive surface so things can move past one another. However, they have many different functions. They protect against invading pathogens, like all barriers, all epithelial-like barriers. They secrete surfactant-like materials, proteoglycans, glycosaminoglycans, to provide the slippery surface, as I talked about, so that the organs can slide past one another and that the organs don't stick to the body wall. They transport fluid and particulate matter through the cells, either through vesicles within the cells, or they can separate their tight junctions. They form lots of junctions, and these particulates can go through. They're involved in the coagulation process, but also in fibrinolysis, and they're very important. If you've got injury in the peritoneal cavity or the pleural cavity, you've got a lot of fibrin deposited, and to prevent adhesions forming, we must break down the fibrin. The mesothelial cell makes plasminogen activators, which convert plasminogen to plasmin, and the plasmin breaks down fibrin. They secrete a whole array of inflammatory mediators. They act as sentinel cells, cells which sit there looking for things that go wrong, and when they do, they secrete these inflammatory mediators so that it recruits inflammatory cells and immune cells to the site of injury. They also secrete a range of cytokines and growth factors for their own repair and the repair of subserosal cells. They can transport leukocytes through what they call stomata. These are openings directly into the lymphatics, so you've got mesothelial cells which are bunched up, and cells can enter into the lymphatic system and be carried away. They can also present antigen, so they have an immune function. Although they're not professional antigen-presenting cells, they can do it. And they may also be involved in prevention of tumour cell adhesion. The reason is that they have a molecule on the surface of tumour cells called CD44, which is a receptor for hyaluronic acid. And mesothelial cells make hyaluronic acid. And it's thought that possibly the hyaluronic acid made by these cells binds to the CD44 molecules and prevents them from attaching to the serosal surface. So it prevents this adhesion and initiation of tumours on the actual serosa. But when things go wrong, or what should I say, what can go wrong? Lots of things can happen that can lead to injury or activation of these mesothelial cells. Surgery, okay, most obvious. Infection, dialysis, Particulates, for example, in the case of asbestos, induce an injury and a repair process. Cancer can also stimulate activation of the mesothelial cells. Possibly indirect cell-cell communication, that is, one cell can communicate to another cell to then activate it. And there's probably many others. Now, what are the model that I use for investigating the repair of the mesothelium, or one of the models we use, is we use a heated metal probe and we apply it to the surface of a rat or mouse testis. And this removes the mesothelial cells in that area on the testis without creating a lot of subserosal damage. You don't get bleeding, etc. So we can keep an intact testis and just look at what effect the injury on the surface does. And if we have a look at this, 2 days after injury, we see the injury is over here, and at the edge of the wound there's lots of cells. A lot of these fan-shaped cells, long or fan-shaped cells, are mesothelial cells that have been activated and have become plump. But there's also lots of inflammatory cells, macrophages, neutrophils, lymphocytes, etc. At the center of the lesion, after about 4 days, you see mesothelial cells creeping over all the inflammatory cells on the wound surface. And by 7 days, you can see there's an intact mesothelium formed. But compared to the photo I showed you initially with lots of microvilli and no discernible cell boundaries, now you can see all the individual cells as they occur. Over time, they will then spread out and will no longer see the boundaries again. And if we look by transmission electron microscopy, here we see the regenerated mesothelial cells, microvilli, tight junctions, etc. And here we have a whole range of other cells which these cells are now sitting on. Now, I want you to think about what induces the activation of these mesothelial cells. This is the picture I showed you before, where you have mesothelial cells right next to inflammatory cells. And many of these inflammatory cells are actually macrophages. If we look by autoradiography, this is where the animal has been injured and injected with tritiated thymidine. Half an hour later, or whatever period of time later, the animals have been killed. And then we do autoradiography, and that means that where a cell is making DNA, you get silver grains above the nucleus. And you can see at the edge of this lesion, we've got dividing cells. You can also see some of the seminiferous tubules are starting to, cells within them are dividing. And if you do a monolayer imprint, we take a gelatin-coated slide, we take the testis and we apply it to the surface of the slide and strip off all the cells, and then do autoradiography after the animals have been injected with tritiated thymidine. We can see here a lot of the dark spots are the nuclei which have got silver grains, so these are dividing. The lighter ones are the unlabeled nuclei. You can see that surrounding the lesion there are a lot of proliferating cells. And what we did is we took these inflammatory cells, we created a lesion in the peritoneum and we washed out all these inflammatory cells. We put them into the testicular sac and stimulated the mesothelium to see whether or not these inflammatory cells could induce cell proliferation. And what we see compared to control medium, fixed cells or control thymocytes, peritoneal exudate cells, and these are all different exudate cells, not just macrophages, neutrophils, lymphocytes, markedly stimulated proliferation of these cells. And you can see that's the image that we took with the positive stained cells. And when, I won't show you the data, but what we also went to show is that this proportion, these cells are predominantly macrophages which stimulate the proliferation of these cells. Now, very interestingly, mesothelium is very different to other epithelial-like surfaces. Epithelium heals normally by centripetal migration. Cells at the edge of a wound move to the center of the wound. And if that occurs, you would expect a large wound to take much longer to heal than a small wound. But mesothelium is different. The time required for small or large wounds is about 7 to 10 days, irrespective of the size. So that means that cells are not just coming from the edges inwards, they must be coming from somewhere else. And this diagram here suggests possibilities. These are two serosal surfaces. The parietal surface is that on the body wall, the visceral surface is on the organ wall, and we have an injury in the middle. We know cells at the edge can migrate onto the wound surface. That's centripetal migration. But some other possibilities are that you may get desquamation of cells from opposing surfaces which become free-floating cells, and then they may land on the wound. Alternatively, there may be a population of free-floating cells there anyway, which can then land on the wound. You could have subserosal fibroblast-like cells. Remember the cells I said to pay attention to at the beginning? These fibroblast-like cells may differentiate and move to the surface to become new mesothelium. Or you may have a circulating cell. So it may come from the blood, from the bone marrow, and then that may enter and come to the surface and become the new mesothelial cells. Lots of different ideas, possibilities. The most accepted idea is that they come from these subserosal fibroblast-like cells. However, in numerous studies that I've performed, we cannot find evidence for that. And most of the evidence that we find is for a free-floating, circulating, or free-floating mesothelial cell. Not sure where they come from, but we do know there are free-floating mesothelial cells, and I'll show you some data to suggest that they can attach to the surface of the wound. One of the reasons why we think these cells come from the edge and float and land on the wound is that at the center of the wound, before we see any cells at the edge moving forward, we see mesothelial cells even at day 2, right in the middle. So they've come from somewhere. We also know if we take pericardial fluid and collect the cells and stain them with HbME1, which is a marker for mesothelial cells, that we see lots of mesothelial cells in the serosal fluid. There's one problem with this approach. It's good and bad. In the process of taking the cells, you sometimes strip some of the mesothelium from the pericardial wall. Hence, you see these sheets of mesothelial cells. But you also get individual mesothelial cells. And we've done several studies in different systems, different animals, to show the same thing. So you do get free-floating cells, and they're viable. One of my honours students, Aina Hoy, what she did is she took a rat and she injured the peritoneum of the rat, and she looked at the number of mesothelial cells in the peritoneum after she washed out the peritoneal cavity up to 3 days after injury, and she showed the percentage of mesothelial cells increased up to day 3. But more importantly, the total number of cells increased up to about 100-fold by day 3. So although we've got a 2 to 3-fold increase here, you can multiply that by 100 to show how many mesothelial cells there. And most of these cells are viable. When I was working in London, one of my PhD students, Adam Foley-Comer, did a study where he cultured mesothelial cells, normal mesothelial cells, and he stained them with a dye called dye I. This stain labels the cells, and it can't be transferred between cells. He took the cells, put them into a solution, injected them into the peritoneum of a rat, injured the testis the way I showed you before, and then he took the imprints, as I mentioned, and looked at them by confocal microscopy. And I'll take you through these diagrams. This is what the cells look like in culture when they're stained and looked at by confocal. 5 days after injury, if we look at the imprint, we can see there's lots of red dye-I labeled cells on the surface of that injured mesothelium, showing that the cells we put in there have now attached to that surface. When we did the same to fibroblasts, we saw no fibroblasts attaching, so it was specific for mesothelial cells. We also took lavage cells, that is all the peritoneal cells, and did the same thing, and we also see themselves sticking to the surface. Of course, there's mesothelial cells, there's macrophages, there's all different cells there. So to eliminate the possibility of these being macrophages, we did another study where we labeled macrophages with another dye called PKH26PCl. This is what they look like in culture. And we did an imprint the same way, and we looked for macrophages on the surface, and we could see some at day 2, the odd one at day 5, but by day 8 there were no macrophages labelled sitting on that surface. So that suggests that these lavage cells are predominantly mesothelial cells. Now what we also showed, and I'm not going to have time to show you here, is that these cells are viable and they are proliferating. If we take a transverse section of the image that I showed you before, we can see here, this is the surface regenerating mesothelium. You can see dye-I labelled mesothelial cells. But interestingly, we can also see cells that are dye-I labelled sitting beneath the mesothelium, trapped. What happens to these cells? Where do they go? That made me think of studies I did many years ago during my PhD, where I was looking at transmission electron microscopy of regenerating mesothelium. After 15 days, here's our regenerated mesothelial cells, microvilli, and we see all these other fibroblast-like cells sitting beneath. Most importantly, the original basement membrane is way down here. So where have these cells come from? Could these cells have been mesothelial cells trapped beneath the new mesothelium, and because they're in a different environment, no longer polarized, differentiate to form fibroblasts or fibroblast-like cells. So that led us to thinking about the possibility that mesothelial cells can differentiate, undergo the process of epithelial mesenchymal transition to become different cell types. And in fact, that's what we showed. Another one of my students did a study where he took cultured mesothelial cells, which look very cuboidal when they're cultured. He stimulated them with transforming growth factor beta, an agent known to induce EMT, or epithelial mesenchymal transition, and he made these cells become fibroblastic. So we can convert epithelial-like mesothelial cells to a fibroblastic form under the process of EMT. So that also led us to think, well, if mesothelial cells can become fibroblasts, can they become other cells? Can mesothelial cells be a progenitor or even a stem-like cell? So one of my past PhD students, who is at this meeting, Sally Lansley, is now a postdoc working within our institute, did a study where she took cultured mesothelial cells and incubated them in the presence of medium which is designed to make cells differentiate into osteoblasts, bone-type cells. And this is the process they use for stem cell differentiation of bone marrow cells. And what she did is she cultured them for various times and looked for markers of osteoblast differentiation. And one of those markers is alkaline phosphatase. And this, you can see here, are mesothelial cells that have been stained for alkaline phosphatase after 18 days in culture with osteogenic medium. You can see they now express this alkaline phosphatase. This is not specific for bone, but it's highly suggestive. And we can see over time we get an increase in alkaline phosphatase. But more importantly, these mesothelial cells will form nodules. These are mineralized bone-like nodules after about 18 to 27 days in culture. And this is indicative of their differentiation to osteoblasts. And here is a bone marrow control cell doing the same sort of thing. So we then showed that mesothelial cells have the ability to differentiate into osteoblast-like cells. But we wanted further evidence. So what Sally did is she looked at a whole range of markers for osteoblasts, CBFA1, osteonectin, osteopontin, bone salloprotein. This is PCR. These are our control bone marrow cells. These are our mesothelial cells. ROS are a osteosarcoma cell line, used as a positive control. And effectively what she showed is that they express the markers for osteoblasts. And the bone-specific one is called CBFA1. It's the only specific one. And she showed that went up over time in culture. And this basically is just the protein. So what we've shown here mimics what we see with the PCR. And this is Western blot to show that the protein also increases. Do these cells undergo EMT? We tested this by looking at a range of markers: cytokeratin 19, E-cadherin, N-cadherin, snail. CK19 is an epithelial marker, as in E-cadherin. It should go down. N-cadherin should go up. Snail should go up. What we showed was a downregulation of E-cadherin and a downregulation of CK19, which is consistent with EMT. In the case of these cells, though, they reformed E-cadherin, and that is because osteoblasts also have E-cadherin in them. So they go through a process of differentiation and then re-expression. N-cadherin remained constant all the way through. And snail was absent at day 0, but increased over time. So again, these cells are showing evidence of EMT. We showed that they can differentiate to osteoblasts. What about another cell type? What about an adipocyte? So again, Sally cultured mesothelial cells, this time in the presence of adipocyte medium. And we looked for the presence of oil red O, a marker of lipid formation. And what she showed was that after a period of time in culture, mesothelial cells started to make and express lipid. And again, this shows lipid increasing over time in culture. And a number of markers, these are PCR for different markers, and Western's showing adipocyte markers, PPAR gamma 2, adiposine, AP2, LPL, and CEBP alpha. And again, they increased over time, showing that these cells can differentiate into adipocytes. So what we showed then, or Sally showed, was that mesothelial cells may be a multipotent cell. They have the ability to differentiate. Maybe they come from the bone marrow. We don't know yet. That's still a big question mark. But you get these free-floating mesothelial cells that can participate in the repair process. These cells can also, when activated, maybe move down into the subserosa and become other cell types, differentiate to myofibroblast, endothelial cells, smooth muscle cells, and also, as we've shown now, osteoblasts, adipocytes, possibly also chondrocytes and neuronal cells. So you can see that mesothelial cells are more than just a cell which induces a slippery surface. They have many different functions. One of them may be as a progenitor or stem cell, which may be important in the repair process, not only for themselves, but in the whole serosal cavity. Now, how does this relate to mesothelioma? We know in mesotheliomas we see bone, we see skeletal muscle, we see fat, and the theory is that these cells can differentiate to form these structures. And although I'm not showing you the data, what Sally also did is take mesothelioma cell lines and do the same thing, convert them to osteoblasts to make bone and to adipocytes. So we know that not only do mesothelial cells, but mesothelioma cells can also make these different structures. So I'd just like to acknowledge the people that were involved in this study. Sally did all the cell differentiation work with help from Cecilia Prell, who co-supervised her PhD, and several others in the lab, and then our lab head, Phil Thompson. And at the University College London, some of the early work I showed you with the cell attachment studies were done by Adam Foley-Comer